Dr. Ahsan Majeed. Today lecture is basically pharmacology, and this pharmacology is again the first lecture is very important introduction to pharmacology on my channel Biology Hub. But inshallah very next that is a complete lectures of the pharmacology with different techniques and tools, and there's a very difficulties, you know very well, the student feel very difficulties in the pharmacology mechanism of action, very difficult to remember the pharmacology. I can give the easy tips, easy way to understand the pharmacological tips and different slides. If anyone student contact me, then I will send the different slide over here that can easily remember the medicines. Okay, first of all, today we discuss the pharmacology that is a penicillin G. That one is a penicillin G. This one is a narrow spectrum antibiotic. This one is a narrow spectrum antibiotic. There is a different spectrum, broad spectrum, and we can say that penicillin G, penicillin V. There is a lower third class that is a basically we can say that is a amoxicillin, ampicillin, ticarcillin, piperacillin, other different antibiotic over in the market and in the pharmacology book. We can study this lecture from the pharmacology pharmacology Katzung and different multi books where I can use over here. So first one, that one is this is the narrow spectrum antibiotic. First we can remember that this one is a narrow spectrum antibiotic, and this is primarily to the we can say that primarily concerned with the gram positive bacteria and somehow others bacteria gram negative. And maybe what is the mechanism of action? What is mechanism of action? This is very important. It acts by inhibiting synthesis of peptidoglycan layer, of which there is a bacterial cell wall. You know that is a very basic concept. The cell wall of the bacteria is made up of peptidoglycan. So the first of all, it acts by inhibiting synthesis of peptidoglycan layer, of which bacterial cell wall. How? By blocking the transpeptidase enzyme activity. It can block the transpeptidase activity, and this activity is blocked, then we can say that bacterial cell wall is burst and bacteria dies. How the bacteria is dies? What is the exact mechanism? What is the role in the gram positive? What is the role in gram negative? We can see over here. This diagram is for the gram positive, we can say that, and this one is for the gram negative. One of the gram positive and one of the gram negative. So what is the basic difference between the, you can see over here, and you can first examine that which one is basically a difference, basic difference. We know that this one is a cell wall and this one is cell membrane. And in the same way, in the gram negative, this one is a, we can say that is a cell membrane, and this one is the cell wall, and cell wall we can say that this, and additional this one is a outer layer. This one is the additional outer layer. So now we can discuss the gram positive and gram negative. So what is the role of penicillin in this? We can say that in this lecture we can see over the penicillin view, penicillin, how it can be react and how it can be produced. In last I can show you basically a different way of the medicinal effects of the penicillin. So first of all, you can see over here that is a outer layer, and in this outer layer we can say that here we can see the porins. Porins is protein, that is extracellular protein. You can say that is a extracellular proteins over here. This one is extracellular proteins, and in the inner layer this is a middle layer that is made up of peptidoglycan. This is a cell wall, and this one is the outer layer. We can see over here, this one is a peptidoglycan. And we can also know that peptidoglycan word is peptide. Peptide means fragments of amino acid and sugar. There is, for example, like we can see in that is a NAM, NAG, N-acetyl glucosamine, N-acetyl. There's a different types of the name over here that can amino acid link with each other with the help of enzyme. This enzyme is known as a transpeptidase. Transpeptidase enzyme. Transpeptidase enzyme. And in the innermost, we can say that in the cell membrane there is a protein, and these proteins are penicillin binding proteins. Penicillin binding proteins. Penicillin binding proteins over here. And what is the actual mechanism? We can say that there is a penicillin. First of all, this penicillins come over here and can react. In the gram positive, there is a no layer, so that's why that can easily react over here. But in the gram negative, we can say that this one is attached on the outer additional layer, then move toward the cell wall, peptidoglycan, and inner pass to the cell membrane, plasma binding protein. So listen over here, this is the mechanism. Penicillin undergoes and plasma bind with plasma binding proteins. First of all, penicillin enter into the membranous system, and this can enter into the plasma binding protein. Once it can attach with the plasma binding protein, it means it activates. It activates. And its activation, after that, it can block the trans enzyme activity, transpeptidase activity. So, first of all, this penicillin move toward the plasma binding protein, activate. Third one, that can block the transpeptidase activity. It can block it. It can do not produce amino acid. It can break the amino acid collapsing. So this can is a amino acid collapse. We can say that bacterial cell wall destroy. Peptidoglycan, it's damaged. How it is damaged? Because transpeptidase activity is blocked. Once it's blocked, it means peptidoglycan is basically damage. So if this is damaged, then we can say that bacteria cell wall destroy, and we this at still end we can say that bacteria dies. So basically one I can just make up this bacteria that is closer to the die, and this one is a mechanism of a penicillin G. So in the same way, in the same tutorials, there's a basics lecture of penicillins and penicillin binding proteins. Come over. Any medicines like regarding to the antibiotic regarding the penicillin, that is the exact mechanism behave like this. So this is my lecture of pharmacology. You can subscribe this my channel, and